So neuropharmacology is the study of how drugs affect cellular function in the nervous system and all the mechanisms, right, all the actions through which they can influence behavior. And drugs, as we were suggesting, often act on protein machines that are involved in chemical communication at the synapse. So I want to go through a couple of these drug receptor interactions with you. So, terminology is rife in any field, but certainly in the field of neuropsychopharmacology. So, two very important terms to understand, though, are agonist and antagonist. So, in this particular illustration here, we have a postsynaptic membrane that is indicated, and we have a number of, receptor targets that are embedded in that membrane. And you can see, cartoons here of a GPCR, right, a metabotropic receptor, with the receptor, sticking out into the extracellular space, right, with a G protein attached on the inside, right? And next to it, we have an example of an ionotropic receptor, right, which is, got a channel that can open. It's gated at the moment, but it can be opened by the binding of a neurotransmitter, right, on the outside in the extracellular space. This is also called, as we described right earlier, a ligand-gated ion channel. Now, what would normally happen if a neurotransmitter acted at the G protein coupled receptor? Here's the G protein on the inside. Here's the portion of the receptor sticking out, the metabotropic receptor sticking out. The neurotransmitter binds here by my thumb. The thing moves, and the G protein is released and can now initiate like a whole bunch of changes inside the cell, right? And here's an ionotropic example, right? Here's the ionotropic receptor before the neurotransmitter binds. The neurotransmitter binds out here in the extracellular space. The thing changes and opens a channel, that lets ions go through, and it can rapidly, depolarize and excite, or it can rapidly, hyperpolarize and, inhibit, depending on the kind of channel and the kind of ion that it's selective for. Well, here we show on the left side here what we call an antagonist drug. And an antagonist is going to bind, to the G protein coupled receptor and prevent the neurotransmitter from binding and releasing that G protein. So it antagonizes, the activity of that GPCR, of that metabotropic receptor. And for the ionotropic receptor, it binds and prevents, the opening of a channel upon, should a neurotransmitter try to get into the vicinity. It can't bind there. It's changed the shape enough so that it won't open properly, even if it does bind. So it's blocking, it's antagonizing. These are antagonist drugs. And on the right side, you can sort of see the opposite. You can see here's a drug that binds to the GPCR, the metabotropic receptor, change in shape. And just like with the neurotransmitter, the G protein is free. It can go, it can leave. And here's the ionotropic receptor. Here comes the agonist drug. Binds, swings open the channel, very much like the neurotransmitter would do. Now, the complexity, of course, arises in that, all of these structural changes with the binding of these various drugs, agonists and antagonists, sometimes you get partial agonist activity. Sometimes you get, partial antagonist activity. We're going to see that while neurotransmitters will bind, to all of their target receptors as, full agonists, basically, they'll just act to activate, the receptors that they're sort of, built to stimulate. So like, as an example, glutamate, has a number of ionotropic targets and, metabotropic targets. Well, glutamate will bind into each one of those, right? But then you can have, for example, some drugs that will, be a partial agonist, partially activate this kind of receptor, but partially antagonize another kind of receptor. So it can be more complex. So these are drug receptor interactions, but you can see antagonists, if it's a full antagonist, it completely blocks the normal functioning of the receptors. If it's a full agonist, it completely blocks the normal, initiation of activity in those receptors.